So, good evening, one and all. With the ongoing paper discussion session, today we will be seeing the 2022 plant science paper, right? So, I think we have dealt with both the 2020 and 2021 genetics plant bits. Now we will be dealing with the 2022 paper. Coming to the next part, the first question is an assertion reason type. Um, So given two statements, right? One is assertion and second is reason. So in case of assertion, it is stated that meiosis is a reductional division. So we all know that mitosis, we say it as the equational division and meiosis as the reductional division. So the main reason why meiosis is considered as reductional division is it changes the ploidy number to half, right? From 2n to n, right? So that is the reason it is given here, that the chromosome number is reduced from diploid to haploid. So meiosis is mostly seen during the gamete formation stage, right? So where the chromosome number it gets reduced to half. So I request you guys to mute yourselves. So with the given assertion, the reason it matches, right? So meiosis is a reductional division, and then the reason is it reduces the chromosome number or ploidy level from diploid to haploid. So with that confirmation, we can generally say both A and R are true, and R is the correct explanation of A, right? And guys, we may also need to remember who gave the term mitosis, who gave the term meiosis, and who described these cell division, etc. And coming to the next question, according to latest classification, prokaryotes are divided into the two domains. Right? So according to the latest classification, it was divided into archaea and bacteria. So it's an direct type of question. And next is other assertion reason type of question. So the assertion is the mode of DNA replication is semi-conservative in nature, right? So can anyone say who are the scientists who proved that the DNA replicates in semi-conservative manner? The scientists who proved that DNA replicates in semi-conservative manner. Anyone? You can name the organism in which they have proved. So you need to know all these things, okay? Right. So they proved in Escherichia coli, Assumption and Stark, right? So these are the two scientists in E. coli. So for proving, they generally use the N15 and N14 isotopes. Right? So we all know that DNA replicates in semi-conservative manner, and the reason given is one of the strands is synthesized continuously, while the other is synthesized discontinuously. So when you go through the DNA replication chapter, it is clearly mentioned that, right, among the two strands, one is a leading strand, the other is the lagging strand. Right? This is what we call continuous and discontinuous replication or synthesis of the DNA. But this reason do not have anything to do with the assertion, even though both are true. Right? It replicates in semi-conservative manner, and the DNA replication consists of both continuous synthesis strand and discontinuously synthesis. So both the assertion and reason are true, but reason is not the correct explanation, right? So this you need to know that who gave the semi-conservative replication, who proved it, who proposed it, who proved it, and in which organism, what are the isotopes. And the other important bit here is what is the gradient they used. It is cesium chloride gradient, okay? This is also important, right? So I hope that now semi-conservative replication, most of the points are covered, right? It was proposed by Watson and Crick and proved by myself and Stahl. And in Escherichia coli, it was a bacterium, and using the N15 and N14 isotopes, and using the cesium chloride gradient. Okay. So coming to the fourth question, so it's just like arranging type of question. So it is mentioned that in the process of mitosis, right? So in the process of mitosis, in a cell consists of a series of events as given below. So arrange the events in order from first to last as per the time of occurrence. So let us see. So the point A states that the highly condensed chromosomes move to the equatorial plate of the spindle. So it is during the metaphase. The centromere, it interacts with the spindle fiber apparatus. So it is during the anaphase, right? So division of the cytoplasm might be at the telophase, and then the sister chromatids move apart to the opposite poles. So even it also takes in the anaphase, and then the nuclear membrane breakdown, and the spindle-shaped structure of microtubules are organized. So it is during the prophase. So based on this, you can arrange the order of events. So as they asked from first to last, so we will be starting with the prophase, as most of you know. So we will go with E, and then we have two anaphase. So if you want to mind, so the first, the centromere, it interacts with the spindle fiber. Then only the sister chromatids can move apart, right? So it goes with B, then followed by, okay, sorry, sorry. So can anyone say that the centromere interacts with the spindle fiber apparatus at which stage? Any answers? You can write in the chat box. Sorry, some background noise. Okay, now is it clear? Anyone? The background noise, now is it clear? If you guys say yes, I can proceed. Okay. So my question was, the centromere, it interacts with the spindle fiber apparatus at which stage? Right? So someone answered it as metaphase. Okay. So we'll move with the metaphase. So we have two metaphase here. So E, then it is followed by. So before interacting with the centromere, all the chromosomes, they need to lie on the equatorial plate, right? So it will be followed by A, then followed by B, right? And then D and C. So this will be the order. Okay. And coming to the fifth question, the direct question, the inflorescence of the rice is called. So we all know the inflorescence of the rice is panicle. And then which of the following amino acid starts all protein synthesis? So we know that the amino acid which starts the protein synthesis is methionine. So can anyone say the code for methionine, the genetic code for the methionine? What is the genetic code for starting the amino acid synthesis or protein synthesis? We know methionine is the compound, but what is the genetic code for methionine? Right. So the genetic code for methionine is AUG, or it can also be ATG. And the chloroplasts are present in the cells of. So the chloroplast, the cell organelle where the photosynthesis takes place, are present in which of the following cells? So in case of fungi, you can't see chloroplast, whereas algae you can see the chloroplast. In plants, you can see the chloroplast. In animals, you don't see the chloroplast. So we have B and C. So it's our B and C, right? So in case of algae and then plants, okay, both these cells have the chloroplast. And then coming to numerical aberration type of question. So there is an terminology. So double monosomy. We know that monosomy is a condition of loss of a single chromosome, but here it is given the double monosomy, so 2n minus 1 minus 1. So trisomy is an condition where we have an extra chromosome present, so 2n plus 1. Nullisomy, where we have both the homologous chromosomes lost. So tetrasomy is a presence of an two extra chromosomes, whereas in case of double tetrasomy, you will be having 2n plus 2 plus 2. Okay, so these are the different conditions. Based on this, you can match them. So arrange the following parts of triticum aestivum in. What is the ploidy level of a triticum aestivum? No, I'm asking what is the ploidy level of triticum aestivum? Hexaploid, right? So we need to represent it as 2n is equals to 6x is equals to. So what is the chromosome number then? If it is in hexaploid, so what would be the chromosome number of triticum aestivum? So it is 42, right? Okay. So we all know they generally give some cells. So endosperm, its ploidy level is 3n. So the male gamete, it is n, and embryo, it will be 2n. So based on this, they have asked to arrange it on the descending order, right, from the highest to lowest. So if you arrange it, you will be having 3n, 2n, and then n. So it would be ACB. Coming to the next question, the genetic codes are degenerative in nature, right? So this is the one of principle of genetic code, and then the degeneracy is observed primarily in the. So where do we recognize the degeneracy, right? So let us consider this is the codon. We all know codon is a triplet, right? So first base, second base, and third base. So always we observe the degeneracy at the third base of our codon, right? And this is nothing but said to be the wobble base position. So the third and then the wobble base position is where we generally observe degeneracy. Okay. So who proposed the wobble hypothesis? Here the question relates to degeneracy and wobble position. So which scientist has proposed the wobble hypothesis? Right, the wobble hypothesis was proposed by Crick. And then coming to next assertion reason type of question. So assertion is the complete absence of false negative results, right? So the complete absence of false negative results would make a diagnostic kit highly successful, right? So just with the help of some logical sense, we can say this is a correct statement, because if there are no false negative results, so the diagnostic kit, it is highly successful, means it is right. Whereas the reason is the false negatives are due to poor sensitivity of the phenotypic and molecular marker deployed, right? So false sensitives are only seen when we have less sensitivity of the markers that we are using. Okay, so both the assertion as well as the reason are right. So this is also the correct explanation. Only when there is poor sensitivity, this is the reason why false negatives occur, right? And if the false negatives are absent in our PCR kit, or in case of your RT-PCR, or whatever might be the technique you are using, so then we can say that the kit is highly successful. So both the statements are correct, and the reason is a correct explanation to your assertion. Okay. So here comes the other question. So suppose you wish to combine the mitochondria from species A, right? So let us write here. So mitochondria, we need to combine it from species A with the chloroplast and nucleus of species B. Okay. So we need to combine the nucleus of species B. So then, what is the technique that we generally use? So here are the given options, but we all know that whenever you need to get mitochondria from any of the plant or any of these cells, so always you need to use that cell or that plant as a female part, right? Only then you can get the mitochondria. So mitochondria, we all know the mitochondrial inheritance or cytoplasmic inheritance is similar to that of your plasmic inheritance, right? So whenever you want to get the mitochondria from species A, so you need to take the cytoplasm from this species and then you need to fuse the cytoplast of the species A with the protoplast of the species B, right? So we need to have the cytoplast of the species A and then we need to fuse with the protoplast of the species B. Okay. And the next question is an assertion where atavism refers to reappearance of a trait after it remains hidden for several generations, right? So this condition or this phenomena, we call it as atavism. And then the reason that they are giving is the chance of combination of genes allows, with the chance of combination of different genes allows long suppressed hidden traits to reappear and express. Okay, so whenever we do meiosis, there is a recombination taking place. So the genes, they get combined in different manner, okay, in different fashion. And with this different fashion, sometimes the long suppressed hidden traits, they have they are been expressing and they reappear sometimes. Okay, so both assertion and reason are correct, and this is the correct explanation of it, right? So can anyone explain, tell me what is an example of the phenomena atavism? Any example to atavism phenomena? Any example? Any common example? Not only in plants, a broad example. Okay. So one such example of atavism, it includes your yeah, appearance of tail. Okay, in human species, reappearance of tail. Yes. Yeah. Okay. So moving on to the next question. So the given assertion is that the DNA polymerase can synthesize the DNA in the 5' to 3' direction. Okay. So this is the first assertion, and then the can synthesize DNA in the 3' to 5' direction. Okay. So we all know DNA, it gets synthesized in only one direction, right? Means in this fashion from the 5' to 3'. But DNA polymerase does not have the ability to synthesize from 3' to 5'. So only the assertion is right in case of this question. So only A is true, but our reason is false. All right. And then coming to the next question. So as per general model, the orders of activities in DNA replication in Escherichia coli from beginning to the end, right? So we need to arrange it from beginning to end. So let us find what is the order. So we all know first the DNA gyrase and then the DNA helicase. They come, they unwind the double stranded DNA. Okay, right? So C is the first. Then after unwinding the double stranded DNA, we need the single stranded binding proteins, right? To make the two single stranded DNA in the separated form. Then followed by B. And then the enzyme primase, right? The enzyme primase, it comes and synthesizes the RNA primer. All right. So do anyone know what is the gene that produces the enzyme primase? What is the gene that produces the enzyme primase? In case of prokaryotes, the gene which is responsible for production of the enzyme primase. Right, it is DNA G. Okay, so do have a look on it. So it is your DNA G, and then helicase it is DNA B. Okay, all right. So both these genes, the DNA G and DNA B, they together are known as primosome. Right. So please note down this. Someone might be knowing this, but both the DNA B and DNA G are known as primosome. Right. So anyone want to ask anything? MSB. Hello. Okay. Right. So the DNA B and DNA G, DNAs actually. So these together are known as your primosome. So next B, after B you will be having the primase to synthesize the RNA primer, and after which the synthesis of the DNA strands goes continuously in the leading strand that we discussed earlier in the same paper, and discontinuously in the lagging strand, right? So followed by E. And then ultimately what happens is after digesting the RNA primers, right, in case of your lagging strand, so the RNA primers, they goes off, and then the Okazaki fragments are present, which are joined by enzyme known as ligase. Uh, this will be D. So this is the order in which the DNA replication takes place in Escherichia coli. And then coming to next question, which of the following characteristics is not true for genetic drift, right? So this is a direct question. In case of genetic drift, so genetic erosion, like genetic drift is not similar with genetic erosion. This is not true. Okay. So genetic drift as well as your mutation, migration, right? All these are exceptions to the Hardy-Weinberg law. I hope you know that the exceptions to Hardy-Weinberg law is genetic drift, mutation, migration, and then there is an other factor. What is it? Anyone? Genetic drift, mutation, migration, and what is the other thing when we are discussing about Hardy-Weinberg equilibrium? Anyone? Mutation, migration, genetic drift, and what is the other factor? Right, selection. Okay, so these are the factors we will be avoiding when we are discussing about Hardy-Weinberg law. And then, so the formation of a loop during the packaging stage of meiosis. Okay, so formation of a loop during the packaging stage of meiosis indicates the occurrence of. All right. So have you? Most of you have. You saw that. So whenever there is a translocation in the packaging stage of meiosis, what would be the configuration? Anyone? What is a configuration when there is translocation phenomena in the packaging stage of meiosis? How the chromosomes would be looking like in case of translocation? Any idea? This is an important bit. What is the shape of the chromosomes? How they look like, right? So they look like in a cross or plus configuration. Okay, so they look like this. So someone call it cross, someone call it plus. And except translocation, in every other stage, in every other these structural aberrations, we generally see a loop forming. Okay, so B, C, and then D. These three, whenever there is a structural aberrations of deficiency or inversion or duplication, we generally see the loop formation. Okay, so B, C, and then D. Where is the option? This. Okay, this is the one. And the next question: if Yeah, we discussed, right? So these are the scientists who proposed the semi-conservative replication of DNA, and they said that the DNA replication is continued up to sixth generation. Okay, so it has continued up to sixth generation in bacteria. So what is the ratio of heavy strands, hybrid, and then the light strand containing DNA in the sixth generation? So simple, you will be starting with the heavy strand, right? In the next generation, you will be having the medium type of strands, right? 1 it gets to 2, 2 gets to 4, 4 gets to 8, 8 gets to 16, and 16 gets to 32. So this is first, second, third, fourth, fifth, and sixth generation. So in case of your sixth generation, totally you will be having 32 DNA molecules, right? So based on this logic, you can easily solve this question by adding the total molecules. So 32 is possible only in this case, right? 0 plus 1 plus 31. So you'll be having 32 DNA molecules. And then ratio, just with the help of semi-conservative replication, you can easily solve the ratio, right? And then coming to the molecular technology, so the expansion of RFLP, right? So we know that RFLP it stands for restriction fragment length polymorphism, right? RFLP is restriction fragment length polymorphism. So whether RFLP is dominant or a codominant marker, any idea? What is the dominance and in case of codominance? So whether RFLP is a dominant marker or codominant marker, what is its nature? So RFLP is a codominant marker, okay? So it was the first molecular marker system before PCR to be discovered, right? So it does not require any PCR. It just requires the restriction enzymes to perform the RFLP and then combined by Southern blotting. Okay. So do remember RFLP requires restriction enzymes along with Southern blotting technique in order to perform this. And then it is a codominant marker. Okay. So what are the bonds that are involved in protein structure? Okay, so we all know the basic bond in case of between two amino acids is your peptide bond, and you can also see the disulfide bonds, right? In case of sulfur-containing amino acids. So what are the sulfur-containing amino acids? Any idea? Which are the sulfur-containing amino acids? Right. So you will be having cysteine and then other amino acid which one, right? So these are your sulfur containing amino acids. In case of protein structure, so when you go from primary, secondary, tertiary, and quaternary, you will be seeing all these kinds of bonds. Okay, all the bonds: peptide bond, hydrogen bond, ionic bond, disulfide, and hydrophobic interactions. All these bonds are seen in protein. Right. The next question is, to resolve a parental dispute, the honorable judge ordered DNA fingerprinting of the child. So the probable DNA fingerprints of the child would be. So this is simple. So the child will be having 50% of DNA from either of your mother and then the father, right? So 50% of the DNA from either of the parents. Okay. And then ribosomes. Ribosomes are made up of. So we know ribosomes, they are made up of both RNA then followed by your protein component. Okay. So ribosomes are made up of both RNA and protein. And the next question is also related to ribosomes. The subunits of ATS ribosomes. So we know ATS ribosomes are only seen in case of eukaryotes. So it is made up of two subunits. One is 40S, then followed by 60S. Whereas in case of your 70S ribosomes, which we see in both prokaryotes and eukaryotes, so it is your 30S and then 50S, right? So here the option would be 40S and 50S, where it is RNA and protein. Okay. So this comes to the conclusion of genetics questions. right? Okay, someone wrote, sir, 0 and 1 and 15 is right for stall. Let's go to that question. So here it has clearly mentioned that they have continued the experiment up to sixth generation, right? So in case of first generation, you will be just having N15 type of strands. Second generation N15, they replicate in semi-conservative manner, right? So from one DNA molecule, you got two, and in the next generation again, this gets divided. So here N14 comes. So this is your third generation where you will be having four DNA molecules. This four, it again gets doubled. So in the fourth generation, you will get 8. And then in the fifth generation, you will get 16. And then in the sixth generation, you need to get 32 DNA molecules. Okay. No means if we divide by 2, then 0, 1 will be right ultimately. No, I can't understand your logic. You can just unmute and clarify what you are telling, okay? And just unmute. Yeah, continue.
Hello, sir.
Yeah, yeah.
Yes, sir. In Nicholson and Charles experiment, sir, 32 strands are, we are getting 32 strands after sixth generation of gene replication.
Okay. Yeah, yeah, strands.
Yes,
Okay, strands are 32. Right, you are right.
yes, sir. And sir, we get two hybrid strands in those 32.
Yeah, we will be getting two hybrid strands.
Yes,
No, I
sir. And
think
sir,
we will be getting ultimately, see, in case of third generation, we have the hybrid strands here. Even in the fourth generation, you will be having the hybrid strands here. Fifth at last, and sixth also at last. So there are two.
yes, sir.
Hello.
So, sir, if we, yes, sir.
Hmm.
Yes, sir. So, two hybrid cells we are getting. That means that two DNA will be there in which we will get one in 14 strand and one in 15 strand. And sir, in the rest of the 30 DNA, we will be only getting sir the in 14 and in 14 strands in both of the strands. So, sir, we are getting two hybrid DNAs among the 32 and 30 are only live. So, sir, um, the ratio 0 is to 2 is to 30. 30.
Okay. Right. So 0 is to 2 is to 30.
30.
Right. So it
Yes,
will
sir.
become
And
1
sir,
is to 15. Yeah. In case of strands, it is correct. Right. So they have mentioned strands. This would be
yes,
right.
sir, I
Okay. Okay.
think the option is because
Yeah. You are right. In case of strands, it is 0 is to 1 is to 15. In case of molecules, we will be ultimately getting 32 DNA molecules. Right.
yes, sir.
Okay. Okay. So I'll just correct myself. All right. So in case of strands, your ratio will be 0, 2, and then 30. If we simplify, this would be the answer. All right. Okay. So up to genetics question, are there any other queries? even now I'll just come back. Okay, here. So these are up to the genetics questions. Okay, so just look upon them, and if you have any queries, you can just answer. You can just ask me. And sometimes, in case of Wobble hypothesis, na, they will also ask in this type, right? So Wobble hypothesis applies only for the first base of anticodon. Is this statement right? In Wobble hypothesis, it applicable only for the first base of anticodon. Okay. In case of Hubble hypothesis, the statement is it applies to the first base of anticodon. Okay. And then in case of codon, it is the third base. Okay. And this was one tricky question. And I know you guys know about the base pairing, how it generally behaves in Hubble hypothesis, right? Some other confusing questions would be like this. So you need to know about RFLP. This is the first marker system uh, that was identified, and it is restriction, fragment length, polymorphism. It requires the restriction enzymes and southern blotting. So I hope we took a class on this, on the marker system, right? And then the codominant marker system. So what do you mean by codominant marker? Like, so why it is called codominant marker? Any idea? Okay, so codominant marker means it has the ability to differentiate between the homozygotes and heterozygotes. Okay, so this is the main application of codominant marker. So once you learn about the marker systems in detail, then you can proceed, right? Okay, so we move on to plant breeding. Right, the screen is visible, right? In case of plant breeding questions. Okay. No. So can't you see the plant breeding questions? Like the match the following type question, first question. Just me share again. Now is it visible? Right? Is it is visible now? Even now, no. Okay. So let us start with the plant breeding. So it's a match the type of questions. So to be frank, even I don't know this type of question, right? In case of vegetative propagation, you always read about the vegetables and then crop plants, but in case of this, I don't know. We'll just skip this question. If anyone has any idea on this, they can comment upon. I never came across these type of plants in vegetative propagation. So orchids and lily are flower plants, and oil palm is your oil and begonia. Any idea on this question? Anyone from horticulture or forestry? Okay, if no, we'll go to next question. Right. So here the list. So one side these are the crop species, and the second is your inflorescence. Right. So in the genetics itself, we saw that the inflorescence of rice is panicle, as we all know. Whereas in case of maize, we call it as tassel. Yeah, someone just want to ask anything? Anyone? Someone raised their hand. Yeah, you can comment, come on, because I don't know the answers to the above question, the match the following. Sir, option 4. No, 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 not like option. I want, where did you come across that question in case of flowering plants? Okay, no issue. So when we see the crop species and the inflorescence types, so we know rice, panicle, and maize. So tassel, tassel. What is the tassel means in case of maize? The tassel, it is your male reproductive organ in case of maize. So safflower, and in case of sunflower, we call it as head or capital 8. In case of sugarcane, we call it as arrow, right? So based on this, you can just match them. And coming to the next, so which of the following two cultivated species of cotton are spinnable fiber? So do anyone know which of the two following species are mostly cultivated and they are also spinnable? We know Gossypium hirsutum, new cotton, it is cultivatable and it is a spinnable fiber, and then followed by barbendens. Okay, so these two, right? So there was one question in the ear net, that which of the following fiber is a seed fiber? Okay, seed fiber, and to be most particular, the epidermal fiber. So it is your cotton. Okay, please to remember seed fiber, that too with a specification of epidermal fiber, it is of cotton. So what is the ploidy level of Gossypium hirsutum along with chromosome number? What is the ploidy level of this Gossypium hirsutum? Tetraploid. So yeah, tetraploid and the chromosome number is 52. And you also need to know the genetic composition. Okay. So also read about the genetic composition. Right. In case of wheat, we know it is an hexaploid with A, B, and D genome. So in case of cotton, what is the genetic constitution with in case of genome? So how would you represent it in cotton? The 52 is okay. Okay, so it is A, D1, D1, all right. And then coming to next question, consider a cross where some offspring in generation, they have a genotype where all the three genes are in recessive nature. Okay, so they have made some cross, and then in the genotype, there is some offspring which has all the three genes in recessive nature. So, which of the following genotypes can be the genotype of either of the parent? So, if you want to obtain all the recessive alleles in the offspring, the only thing you need to do is you need to self the heterozygote, right? Only in the given options, only when you self this type of heterozygote, then you can find all the in some of the offsprings which have all the genes or alleles in the recessive condition. All right. And then for the notification of variety, the essentially required information are, so if you want to notify variety, what we need to do? So first, we need to identification of candidate entry by respective coordinated project workshop, right? So first the AICRP, they need to identify the entry, and then the AICRP will be going on testing, that is the evaluation of the entry that you have submitted. After everything, so the release of the candidate entry by the variety release committee, right? So both the, either it can be your central variety release committee or state variety release committee, they need to release this variety and then it should be notified. And the next is the distribution of the candidate entry through KVK. So this is not an essential step, okay, but these three are essential steps. For the notification of a variety, we need to identify and then uh, proceed it through a proper AICRP trial. They will be conducting the evaluation, and then after which the central or state variety release committee, they will be releasing the notifications. Okay. Next question. Assertion reason type. So inbreeding depression is the reduced survival and fertility of offsprings of related individuals, right? And the reason is inbreeding depression. It increases the heterozygosity, exposing the harmful recessive alleles in the homozygotes. Right. So when you do inbreeding, so inbreeding it increases homozygosity, but it decreases the heterozygosity. So here the statement is a contradiction. So this reason or statement it is wrong. Okay. So inbreeding it does not increase the heterozygosity. Instead, it decreases the heterozygosity and it increases the homozygosity. Okay. So your assertion is true, but the reason is false. All right. And then next, coming to the next question, the test cross ratio. Okay, the test cross ratio for a trait governed by two genes with masking gene action. So if we first let us discuss about the actual ratio of masking gene action in case of F2. What would be the actual ratio in case of the masking gene action? Anyone? What is the actual ratio? What is the actual ratio of the masking gene action? So if you know the actual ratio of masking gene action, you can easily solve the test cross ratio. So let us write what are the genotypes that are possible in F2. Right. So in case of normal F2, it will be 9, 3, 3, and 1. But in case of masking gene action, these two gets clubbed 12, 3, and then 1. Right. So in case of your test cross ratio, right, you will be getting 1, 1, 1, and then 1. All right. So but these two gets clubbed as similar to that of this thing, and it gets converted to 2, 1, and 1. So this would be your answer. Okay. So any type of the question, it can be easily solved in this manner. Okay. So the masking gene action normal ratio is 12, 3, and 1. So if you convert it to the test cross ratio, it will be your 2, 1, and then 1. All right. And then coming to the next question. So matching question. So we all know, with the help of anther culture, okay, you can, when you do anther culture, then followed by your tissue culture techniques, there might be some variation which we account it to the gametoclonal variations. And then asymmetric hybrid, it is a condition where you will be manipulating with the ploidy levels. Okay. And then analytical breeding. So, okay, no, asymmetric hybrid, yeah, you will be manipulating the chromosomes. Sorry. Whereas analytical breeding is manipulating the ploidy level. So do anyone have an idea who gave the analytical breeding concept? Which one should I explain? The masking gene action question? Okay, chase is okay. So test cross okay. Okay. See, we all know that normal F2 ratio would be 9 is to 3 is to 3 is to 1, right? If there is no epistasis, if there is no gene interaction, the normal F2 ratio. So if you write the F2 genotypes, so broadly they can be classified into these types, okay? The normal ratio is 9, 3, 3, 1, right? So what does your masking gene action states that one of the dominant gene, it has the ability to mask both the dominant and then the recessive genes, right? So let us consider in this example, this the dominant gene A, it has the ability to mask this dominant gene as well as it also has the ability to mask the recessive gene. Only in these two cases, gene A is dominant. So which means these two will resemble the same. So it gets clubbed. Your ratio gets converted from 93312 to 1231. Is it okay? Up to this point, 12 is to 3 is to 1, right? So from 9331, your ratio it gets converted in case of masking gene action to 12 is to 3 is to 1. Is this point okay? Right. So with the same case, if you go to test cross ratio, 1 is to 1 is to 1 is to 1. Normally it is 1 is to 1 is to 1 is to 1. But similarly here, these two gets clubbed, which means it will be 2, 1 and then 1. So now I hope you are clear with it. Okay. Is it clear now? Okay. So coming to the present question. So we know when we do the anther culture, there will be some variations and then we account it to the gametoclonial variation. And then in case of a asymmetric hybrid, we can get some chromosomes, we can eliminate the chromosomes, right? Analytical breeding concept in ploidy manipulation, which was given by Chase, and then actinomycin D, which is related to the virus elimination techniques. Okay. And then coming to the next question, the steps involved in the utilization of hybrid vigor, that is heterosis in maize. Which of the following steps are included? So if you want to produce the hybrid, we need to identify and produce the inbred lines. So this is the first step. And then crossing of the selected inbred in suitable combinations to produce highly productive single cross hybrids, right? So single cross hybrids are the product of crossing the two inbreds, we know it, and mixing different single cross hybrid seeds to produce productive double cross hybrids. So this is one statement. It has been said that mixing the different single cross hybrids. And the second statement states crossing of single cross hybrids in suitable combinations to produce highly productive double cross hybrids. Okay. So instead of mixing them, we need to cross the single cross hybrids based on their performance if you want to obtain an superior double cross hybrid, okay, or a productive double cross hybrid. So the steps follows A followed by B, then followed by D, right? So this would be your option. Okay. So who was the scientist that was responsible for heterosis in maize? Any idea? Scientist who was responsible for heterosis in maize. All right. Shal was the scientist who was responsible for heterosis. And then which of the following constitutes genetic emasculation? Right. So here it is stated that genetic emasculation. So emasculation is nothing but the process of removing the male plants, or we can say it as a synonym of your male sterility, right? So male sterility, which is associated with the genetic nature, right? So it can be possible only in case of your genetic male sterility, transgenic male sterility, and then cytoplasmic male sterility. But genetic emasculation we can't describe for chemically hybridizing agents, that is your CHAs, right? So the reason is if you spray the CHA or your male gametocytes, these are not inherited to the next generation, okay? Up to that generation, the male plants or the male flowers, right? They become male sterile. But in the next generation, no, right? So this is the reason why, in case of genetic emasculation, we just consider the genetic male sterility, transgenic, and then the cytoplasmic genetic male sterility. Okay. So in case of chemically hybridizing agents, these are not heritable, right? And then the next question, the mating schemes that increase the homozygosity. So which of the following increases the homozygosity in plants? So homozygosity, it increases only when you cross or when you mate two types of plants. One is genetically assortive, which means genetically similar looking plants. And then the next possible way is phenotypically assortive. Okay. So only in these two conditions you have the possibility to increase the homozygosity. Okay. And then so in maize the diploid chromosome number is 2n. The number of chromosomes in endosperm, so endosperm is your 3n, pollen mother cell 2n, pollen tube nucleus n, and then the root tips 2n. So based on this you can sort it out. Okay. Right. I would skip this question. This is a pathology question. I'll request the pathology instructor to deal with it. All right. And then, so this is the other simple question. So in case of maize, the opaque 2 mutants are found to have some of the following quality parameters. So which of the following quality parameters are seen in case of opaque 2 mutants? So I would like you to answer this question. Which of the following amino acids are more in case of opaque 2 mutants in maize? Anyone? You can write in the chat box or you can just unmute and tell. So in case of opaque 2 mutants, which of the following amino acids are high, means high in their content or quantity? Still, someone said methionine and lysine, then followed by methionine and tryptophan. Any other answers? Yeah, all the options are lysine, methionine, and now tryptophan and so mostly designed for high. Yeah, now I'm audible, right? So opaque mutants, these are designed for high lysine and tryptophan conditions, okay? For high lysine and tryptophan. But in some books it is also mentioned about methionine, but mostly it is lysine and tryptophan. And coming to next question, for hybridization, in which of the following crops the florets are clipped at the top, right? So the clipping method of hybridization, right? So which of the following crops where we see the clipping method to expose the anthers? Okay. So in which crop this method is followed? It's a direct question. Floret clipping method. We call it as floret clipping method to expose the anthers. Okay, instead of saying the crop, you can mention its scientific name. Any answers? Sunflower. Then what is the scientific name of sunflower? What is the scientific name of sunflower? Helianthus. All right. Okay. So are you sure that the clipping method of florets is followed in sunflower? Barley. All right. What is the scientific name of barley? Someone said sunflower with scientific name Helianthus, and now barley, Odium vulgare. All right. So the floret clipping it is followed in many crops like barley, even in rice, sunflower. So I can't tell which is the correct option here. And then next question. So it is said that the clone is immortal. So what does it means? A clone does not die, or a clone can't be eliminated. The genotype of a clone it does not change over time, and then the performance of a clone does not change with time. So we all know that the main statement that the clone is immortal means the genotype of a clone it does not change. Okay, because it reproduces through asexual reproduction, so the genotype does not change. This is the reason why we call the clone as an immortal. And then coming to the next question, so hybrid. So we know that hybrid, how we develop an hybrid, the first generation cross product that has grown commercially. Alright. In case of a pure line variety, it is the progeny of a single self-fertilized homozygous plants. And then coming to composites. So composite is your any type of variety which are produced by mixing the seeds in equal quantities, okay, of several phenotypically outstanding lines, which are not tested for any combining ability. Okay, we'll just see whether they are phenotypically outstanding. Okay, then we will mix their seeds in equal quantity, right? This is important. Mixing the seeds in equal quantity, right? Which are not tested for any of the combining ability, right? We'll be just seeing whether they are phenotypically superior or not. So such type of varieties are known as composites. And then synthetics are a variety which are developed by intercrossing a number of genotypes of superior combining ability. Mostly we'll be testing for GCA, but in case of here, we say it is said that intercrossing a number of genotypes. So uh, what is a genotype that we use in the development of synthetic variety? Any answer? What is the genotype that we generally use in the development of the synthetic variety? We'll be intercrossing them. Before intercrossing, we are also testing them for GCA, right? So, what is the genotype? Okay. In case of synthetic variety, we will be generally using inbreds, right? Inbreds which have high GCA content, then we will be open pollinating or intercrossing them in order to develop the synthetic variety. So, based on this, you can identify them. And the next question is the Daiichi, it ensures that outbreeding will always occur. All right. So which means Daiichi ensures cross pollination. And then in Daiichi, male and female flowers are present on different parts of the same plant. So this is a wrong statement, right? So this statement we generally call it as monishi. In case of Daiichi, the male and female plants are itself different, right? But the assertion is right. It helps in cross-pollination or outbreeding. But in case of Daiichi, the male and female parents are different, or male and female plants are different. This is in case of Monishi, where we'll be having the different parts on the same plant. So, can you tell the example of Monishi, Monishia's flowers? Example of Monishia's condition. In which crop you can see Monishia's condition? Any idea? Is it now audible? Hello? Is it now okay? Yeah, guys, please tell me. I can't see the chat box. You can unmute and speak, without which I can't know. Okay. So, which was the question that we were discussing? Like before, my voice was not audible. What was the question? Tell me which question we were discussing. Was it the question? Okay, 42. Right. So let me arrange everything and explain you. So this is a direct type of question that stating preceding crop requirement is essential consideration during the seed production, right? So before going for seed production, you need to know which crop was there before. So we need to practice this technique to minimize the chances of getting the genetic contamination, right? So we need to remove all the volunteer plants, okay, the preceding crop, whatever stubbles or any seeds, whatever it might be. So we need to remove them. These plants are your volunteer plants. And then coming to the next question, the use of synthetic varieties is more beneficial than single cross or double cross hybrids for which of the following reasons? Okay, so let us see for which of the following reasons the synthetics are preferred more when compared to double cross and single cross hybrids. See, the yield of synthetic varieties is higher than the hybrids. See, hybrids are the one which have the highest yield. No other varieties can have high yield when compared to synthetic varieties. So this is your wrong statement. And the next statement is farmers can save their own produce as seeds and no need to buy new seeds every year. In case of synthetics, this is right. In case of hybrids, you need to buy the hybrid seed every year. Okay. So that is why we call the seed replacement rate of hybrid is very high. Okay. So the synthetic varieties have wider adaptability than hybrids. Even this statement is true. When compared to hybrids, the synthetic varieties they generally have wider adaptability because they are tested for GCA. Okay, but hybrids are mostly tested for their SCA, and then no need to make fresh crosses every year to produce the seeds. Right? In case of synthetics, no, you need not to combine again the parents to produce the F1. But in case of hybrids, you need to make the crosses in order to produce the F1 seed. That is why you need to purchase the F1 seed every year. You need to make the crosses of the parent crosses every year. Okay. So leaving the first statement, all the three statements are favorable statements for selecting the synthetic varieties or preferring the synthetic varieties over your single cross or double cross hybrids, right? And then the next thing is they have just asked arrange the following in the order of their coming into existence. So we know PPV-FRA, it was passed in 2001, and then CBD in 1992, and then Indian Patents Act 1970, and then UPOV, it was in 1961 in Paris, right? The convention was in Paris. So based on this, you can generally arrange them. All right. So one important point here is, so the PPV-FRA, who was the chairman for PPV-FRA? Any idea who is the chairman for PPV-FRA? Any idea who is the chairman for Lochan Mahapatra. All right. So who was the former chairman in 2022? Who was the former chairman of PPVFRA? Yeah, the former chairman was KV Prabhu. Okay. All right. And then, which is the city or which is the place where a convention on biological diversity took place? In which place the CBD took place? Okay, Brazil is a country, and any location, specific location? Okay, please do read about what is the, um, yeah, it is Rio de Janeiro. All right. And then UPOV convention was at Paris. Okay. And the PPVFRA headquarters, in which city is it located? The PPVFRA headquarters. All right. In Delhi. Okay. Good. So these are be aware about all these acts and then IPR related concepts. Okay. In case of IPR related concepts like when did the TRIPS come into existence, right? When did the WTO, WIPO, all these things. You need to be knowing all these things in case of IPR concepts. All right. So I think this is the last question for plant breeding. Yes. So with this, we'll be completing both genetics and plant breeding. If you guys have any specific doubts, I'll be happy to answer. Any specific queries? Yeah.
Sir,
Yes.
sir, which stage is the physical basis of independent assortment? Is it metaphor or anaphora?
Which stage?
The physical basis of independent assortment.
Okay, independent assortment, it takes place in metaphase.
Uh, okay, sir.
Okay. And many other issues. Like, did the exam date come out?
No, sir.
Okay. So if you have any other queries, you can just ask. Like, you also need to know about the different protocols, Cartesian protocol. There will be different protocols. Okay. And where they happened, monetrial protocol, where did it took place, all these things. Okay. All right then. So if you don't have any other queries with this, I'll be ending the today's session. Thank you all for attending. And even if you have any specific query, you can ping on WhatsApp. Okay, right? So thank you all for attending this session. And if you have any specific query, you can just chat on WhatsApp, right? Thank you.